Hello everyone, I am Mrudula and today's topic is the drug discovery and development. So let's start with the definition of drug. So drug is any substance that is used to prevent, diagnose or relieve any symptoms of the disease or any abnormal condition. Then the second definition is about the drug discovery. So drug discovery is the process of designing and developing new chemical moieties for the treatment of disease. The third definition is the drug development. Drug development is the whole process of bringing out the new product into the market when once a lead compound is found. Now we see the history of drug discovery. Before 1919 there was only the herbal drugs and serendipitous discoveries for any disease. Then in 1920s to 1930s, vitamins and vaccines are developed for the treatment of disease. Then in 1940s, antibiotic era was come and R&D boost due to the World War II. Then in 1950s, new technologies was developed and discovery of DNA is done. Then in 1960s, breakthrough the etiology. Then in 1970s, rise of biotechnology was done and use of IT was started. Then in 1980s, commercialization of drug discovery and combinatorial chemistry was in use. And in 1990, robotic automation was coming rise. Now we see the registration process for drug discovery and development. So the Ministry of Health and Family Welfare and the Ministry of Chemical and Fertilizers have the major role in regulation of IPM, that is Institute of Personalized Medicine. Then NDA must be submitted to the DCGI, that is Drug Controller General of our India. Then phase 3 study reported to the CDL, that is Central Drug Laboratory, Kolkata. Then package inserted by the DCI, that is Drug Council of our India. And finally marketing was done by the FDA, that is Food Drug Administration. Now we see the basic principles of drug discovery and development. So it presents the multifaceted process of identifying the new drug in this new era. Then provide comprehensive explanation of enabling technologies such as throughput screening, then structure based drug design, molecular modeling and the pharmaceutical profile. Now the objectives of drug discovery and development. So first objective is to bring medicines that improves the lives of patient, and another one is it is lengthy and resource intensive process, requires close cooperation across multiple disciplines. Now we see the steps of drug discovery development. So first step is drug discovery and development. Second one is preclinical research. Third one is clinical drug development process. Fourth one is the FDA review, and final step in drug discovery development is the post-marketing monitoring. Now the next is timeline. So there are now the next is timeline. So drug discovery was done on 10,000 compounds. Out of that 10,000 compounds, only 250 compounds are sent to the preclinical trials, and time required for this process is nearly about 6.5 years. After that, from that 250 compounds, only 5 compounds are sent to the clinical trials, and the time required for clinical trials is nearly about 7 years. After clinical trials, the FDA review was done on that 5 compounds, and the time required for FDA review is nearly 1.5 years. After 1.5 years, the FDA finally approves the one drug, and that drug was sent for the post-marketing. Now we see these stages in drug discovery and development. So first one is the target identification. So it is the first and foremost step in the drug discovery and development, and which is the heart of modern technology. So it is the process of identifying direct molecular targets of small molecules such as nucleic acid and proteins. Then another one is the target validation. So it is the critical first step which requires nearly 1 to 6 months to complete. Initial computer modeling to screen targets. then and it is the multi-layered step in the drug discovery and development. Now the third is assay development. So in assay development, animal based in vivo studies, then cell based in vitro studies, and high throughput screening are done. Then the next is screening, which is done for to check the potency and the dose response. Then last one is next one is the lead optimization. So lead optimization refers to the designing and development of pre-identified lead compound. The main goal of lead optimization is to maintain the drug components as it is. So the next is development. So it is the preclinical package. Before testing a drug on people, we have to check that drug is safe and is not giving any harmful effect to the people. So aim of the preclinical studies is that to check safety and effectiveness of drug, and another one is its pharmacokinetic properties. So preclinical studies are done by two types, that is in vitro and in vivo. In vitro means outside the animal experiments are done, and in vivo means inside the animal by introducing drug into their body. After preclinical trials, there are clinical trials. In clinical trials, the drug is directly applied on human. So there are mainly four phases of clinical trials: phase 1, phase 2, phase 3 and phase 4. In phase 1, the drug is tested on 20 to 80 healthy volunteers to check the safety and efficacy of that drug. Then in phase 2, approximately 80 to 300 patients are involved and with targeted disease, and on them also the safety and efficacy was checked. Then phase 3, it collects the data of drug, that is that drug is safe or not. Then phase 4 is the post-marketing surveillance, in which FDA approval for a completed drug application, and it is done when the drug is marketed. Thank you.